All right, thanks. Welcome back to the second session. I'm one of the generalists for the day, but in some sense, maybe you can imagine that the eco-evolution day, which is technically tomorrow, starts early. Okay, so I'm going to talk about ecology and evolution in high dimensions, and I talk about what I mean by that. There's a chance that you might hear a second talk of that type tomorrow. Okay, but just before I start, I just want to thank all of the people with whom I spent time sort of thinking about what I'm going to tell you today. And these are Kobe, Sarah, Emmy, and Pankaj in various places, and they've really been steadfast collaborators and instrumental in sort of many of the things that I'm going to tell you about today. And so with that out of the way, my goal today is going to be to try and convince you that in some sense, understanding eco-evolutionary dynamics in highly diverse communities as opposed to not very diverse situations is an important and interesting goal. And I'm going to try and tell you that this, I think, is not going to be achieved by a simple extension of the current models or the current theories that we have. And instead, my sense is that it's going to need new theoretical approaches. And I'm going to spend some part of my talk describing to you what those might look like, but then also just posing questions so that maybe we can take them over into the discussions to follow. Okay, so of course, just to start us off with and to orient ourselves, we probably know this, that everywhere we look around us, there are life forms and different organisms, and they all exist as part of these ecosystems. And these ecosystems, if there's one thing that we know about them, is that they maintain incredible biodiversity. And so there's many, many species that you see sort of around you, and then the idea is that all of these species are interacting, and by means of these interactions, they self-organize, and it's only as a result of that you see all of them thriving together. This is true in this sort of macrobial realm, but it's also true in the microbial realm. And it's really, I would argue, in the microbial realm that just sort of allowed us sort of unprecedented quantitative views of ecosystems in general. And this has been in large part due to sequencing technologies that have really developed and become mature in the last decade or two. And so the idea of all of these approaches, just to give you a sense, is that you go out and you pick out any sample. Let's say you want to survey something about what happens in the soil microbiome. You go and you get many, many samples from the soil. You dig in and you sample these soils. These soils will contain many, many different species of microorganisms. You put them through a DNA sequencing machine, and then you sort of analyze and ask what kind of species there are and what sort of abundances that they survive at. And the idea with microbial communities is that you can do this for the soil, but you can do these for oceans and marine systems. You can do these for things that you're growing in the lab. And sort of it's by doing this that we've learned sort of many, many different quantitative and statistical properties of these kinds of communities. And again, just like we've learned in bigger systems like rainforests, we've also learned that microbial communities also maintain incredible biodiversity. So just to give you a sense of the numbers, much like what Krishna was doing earlier this morning for the cell, at a larger scale, what you see is that you have anywhere between 10 million to a billion microbial cells per gram, which comprise anywhere between 1,000 to 10,000 species, depending on how you count species. I don't want to get into this today, but we can have a separate chat about that. And of course, we've learned many, many other things. And so one of the things that has become clear by this property of these ecosystems maintaining this large biodiversity is that maybe the old paradigm of classical ecology, which I would call low-dimensional ecology, maybe it doesn't quite work anymore. And so just, again, to give you a sense, if you open a classical ecology textbook, most of the time will either be spent talking about one species or two species and different techniques of nonlinear dynamics to study what kinds of dynamics of these things that you get. But sort of more recently, what sort of we've learned is that you don't have to worry about what happens for one or two species directly. If it is really that you're interested in the regime where there are many, many species, you can approach that sort of directly using techniques from random matrix theory and statistical physics, some of which you heard Krishna talk about earlier today. And so when I say ecology in high sort of dimensions, these are the high dimensions that I'm talking about. You think about a space where every species is sort of an axis of some sort. right? And so every community becomes a point in this high dimensional space. Just to also give you a sense of what I mean by high dimensional ecology models, there are a few of them, and the ones that I sort of often study are these things called consumer resource models. And the general idea of these consumer resource models is that essentially you're modeling both a set of species as well as a set of resources, and you're modeling the sort of collective coupled dynamics between them. And so you have two sets of equations that you have to worry about: one for the dynamics of species, and one for the for the dynamics of resources. So what I've done here is just to give you a sense. You have species with different abundances, and so species I with sort of abundance n sub I will have sort of dynamics where there's some amount by which the species grows by consuming the different resources that are available in the environment. And so there are in principle some parameters Ci alpha which characterize how this species I consumes resource alpha. And it's by collectively consuming all of the different resources that it gets some sort of growth. And then maybe it pays some sort of maintenance cost or experiences some sort of mortality. And the net growth rate of each species is then sort of set by a sum of these two things. So that's what's happening for species in sort of many of these models. Different models can look a bit different. And then you also have dynamics for some sort of resources. And so with resources, you might have, in this particular model, three types of things that can happen. These resources can be supplied by the external environment at some rate K alpha. They can be depleted by different species as they sort of consume them. And then in sort of microbial ecosystems, often what you see is this other phenomenon whereby species don't just consume different resources, but as a result of consuming different resources, that they then produce different byproducts. And this is a phenomenon called cross-feeding, which then you can also incorporate into these models. So just, again, the details of these models are not so important for what I'm going to talk about, but there's some set of terms that have to do with how different species deplete resources. So they all come with a negative term, with sort of a negative sign. And there's some sort of terms that tell you, okay, if I eat resource alpha, I'm going to produce resource beta in this amount, and that just has to do with this idea of cross-feeding, right? And so when we say these are high-dimensional ecology models, what I mean is that these are actually not just two equations, but many, many equations. So there's one for each species. And so in principle, there can be S such species. There's one for each such resource, and there can be M such resources. And when we're really talking about high dimensions, we're interested in sort of limits or regimes where the number of species and the number of resources go together to some large number. Right? And so in this kind of regime, a natural question that you might ask is, how do you sample all these different parameters that are present in these kinds of models? And thankfully, I don't have to reintroduce this. Krishna already did this for me earlier this sort of morning. And this idea sort of goes back a long way to Eugene Wigner of sort of taking anything that's complex and replacing it with something effectively random. Right? So Wigner was thinking about it in terms of the uranium nucleus, to replace sort of all the different components of what's present in the uranium nucleus with essentially a random matrix. And we sort of try and use the same approach of taking metabolism, which is quite complex, and trying to replace it with essentially a random metabolic matrix, where this matrix will have some notion of which species are present as some rows, and then which resources they're able to eat as some columns. And so then every cell of this matrix would then tell you with what preference the species I consumes resource alpha, right? And so the idea is that you can also then sample the parameters of these matrices using some ensemble that you will choose. Okay, and I mean, this idea is, of course, it's old in physics. It goes back to the 50s, but it's not by any means new in ecology. This idea goes as far back as the 70s, pioneered by Robert May, where he was asking the sort of similar questions about whether more and more diverse ecosystems you expect to be more and more stable. And he had some nice and quite sort of consequential random matrix theory analysis to sort of argue that that was not the case, that larger systems would not tend to be stable. But then I would argue in some sense, since the 70s, this approach was kind of lost to the ages. So, and then so even though the idea of this approach is that you think of species and their interactions with the environment as random variables, much like I was describing here, this idea from the 70s has found sort of a resurgence in the last decade or so. And there's been work done by lots of people, some of them in this room. And really the idea from doing a lot of this, what I call high-dimensional ecology, has had a big influence in how we think on how big ecosystems stay diverse, stable, what kind of dynamics they have, whether or not these dynamics are chaotic or not, and in particular then how niches are structured and constructed by species acting together. The price you pay, of course, much like what Krishna was talking about in his talk, is that you can now only talk about the statistical properties of ecosystems rather than being able to say something very, very specific about this species doing this tomorrow. Right? So you can't make predictions of that kind because, of course, these parameters are random, but you can still say something statistical. For instance, how many species do you expect, much like Krishna talked about how many phases you might expect. Right? Okay, so this is all of this stuff about high-dimensional ecology. You can do a lot. And really, in any other talk that I would be talking about, this is exactly what I'd go on to explain to you. But in today's talk, I want to talk about what this work does not have. And in particular, if you look at and focus on much of this work, what you realize that it ignores this fact of biological evolution. And there's two key sort of mathematical ways in which this work ignores this. And I just want to illuminate them to you and then expand on how they become problems when you want to do not just high-dimensional ecology, but high-dimensional ecoevolution, may I say. And so there are really two key relevant assumptions here. The first one is the assumption of independent species pools, which is to say that normally what we're imagining when we're doing high-dimensional ecology is that you have some parameters that you pull out for species 1, species 2, species 3, and so on and so forth. And usually you'll pull these out independent, which is that whatever it is that you pulled out for species 1, it'll have no bearing on the the physiological parameters of species 2. And this is quite reasonable, right? If I have species 1, it evolved on its own in some way. Species 2 evolved on its own in some way. So maybe, at least as far as the species pool is concerned, you may not expect reasonably to be any similarity between species 1's ability to consume glucose and species 2's ability to use glucose. While this might be reasonable when you think about ecology, and especially when you're talking about assembling species from a big pool, this is not something that you would expect to be true when you're talking about evolution, because there are at least two things, a parent and a mutant, that you would not expect to be completely independent in their traits. And so you would naturally expect parents and mutants to maybe not be the same, but to at least be similar in how they consume resources, do various things, and so on and so forth. And there really are various assumptions when you're doing calculations in high-dimensional ecology, but essentially the central one that this is really a problem for is some version of the invocation of the central limit theorem. Really what you're trying to do in much of this work is you're trying to use the central limit theorem, the idea that is that you're picking many, many random numbers, independent and identically distributed, and then you can say many, many things about their sums and so on and so forth. Well, as you start to get correlations in these sorts of matrices, this is no longer going to be true anymore. This is something that's confusing to you. Hold on to this thought. I'll expand on this as I go on. The second assumption is actually one of deterministic dynamics. So just like the example of the consumer resource model that I was sort of telling you about, those kinds of dynamics, even though the parameters that they had were some quenched random variables, the dynamics themselves were actually deterministic. And you might say this is again quite reasonable when you're talking about ecology. But again, if you start to think about evolution, as we are getting into the mode of doing, this is again not going to be the case anymore, because every mutant, when it starts off, is going to start off at a very low population size, well, typically one individual. And this is the regime where it will naturally have to encounter the stochasticity that comes with small population sizes. And again, if this part, yeah, go on.
like naive question about, I mean, this is also maybe slightly unfair.
Yeah.
So potentially space matters quite a bit as well, right? So how much, I mean, one axis is evolution, but the other axis is space, and that, how much are we missing in the data is one question that I always have.
Yeah, excellent question. So space is indeed one thing that we ignore in much of this work. And I would say this is generally true for most of the work that I was talking about in the past few slides. How much it matters is, I would say, an open question. How much data we have some, but not a lot. So there's some very nice experiments that people have done, at least with 2 or 3 species or strains, showing how spatial structure, especially when there's cross-feeding, can matter to bring clusters of strains that are interacting together. But I would, and then, you know, lots of people talk about what happens when populations are expanding in space, so like range expansions and stuff like that. But again, for the vast majority of the work that I know, it's either with one species or a small number of species. So again, how much we have in terms of data, it's sort of hard. And I mean, on the data side, I would say it's much more understandable because, well, if you have 20 or 30 or 40 different species, then telling them apart just spatially is going to become hard. But I would say even from a theory and modeling perspective, there's not a lot of work on how all of these things change when there's space, and could be actually one thing that we spend some time talking about in the discussion. Yeah.
I have a clarification and a question. One is your second point, the fact that these models typically assume deterministic dynamics, has anything to do with evolution, or is it not?
It has, it has a lot to do with evolution. And actually, if it's not clear right now, it'll become clear in 2 or 3 minutes, because I'm going to spend a bulk of my talk talking about what happens when you have stochasticity and why it's sort of hard to ignore.
Okay, no, the other comment is about this independent species pool. So you were saying that species are related, but the other effect here would be coevolution, right? So
coevolution, yeah, exactly. So in some sense, what I'm also trying to say is that coevolution is not something that you're thinking about, essentially because you're always imagining the process of assembling a community as one where there's like a big pool of species that's been somehow generated by God, and then you're assembling into any patch a community. Yeah, but yeah, so coevolutionary dynamics are also something that people don't study. Yes.
One more question, sorry, a clarification. So evolution also has 2 different time scales of interest.
Oh yeah.
One is one that's genetically modifying the source code to generate variants, and one where presumably these microbes have some physiological plasticity, which might show up as some time-dependent or varying structure to that matrix. So in the limit, you forbid mutations happening.
Yeah.
You could still get that the same molecule has some physiological plasticity. So in your mind, is that something that's accounted for already, or is that something that needs to be, you know, resampled by resampling the random matrix every few?
It's funny because whenever I talk to people who work on cancer a little bit, they always tell me that there's a lot of phenotypic switching and heterogeneity, which is something that you need to worry about. And so the way that I often think about this is that, well, there's 2 ways, one in which it is incorporated and one in which it's not, which is that, well, if there are 2 different phenotypes, there's nothing stopping you from thinking of the different n sub i's as 2 different phenotypes. But the second way is that really, if you wanted to take that seriously, you would also have to add another term which has to do with switching from one type to the other. And that could then in principle be as complex as you want, because then you have to think about all the regulatory mechanisms by which someone decides to switch from phenotype X to phenotype Y. And these currently don't include these at all. In some sense, you could imagine that if those phenotypes were there to begin with in these models, then the fixed points or the natural dynamics just tell you what happens to the abundances of the different phenotypes. But if you really wanted to think about the switching aspect, that's something that's ignored currently. How important it actually is, I do not know. Yes, exactly. So you're imagining that resources are what people call substitutable.
There tension where it becomes important, or?
Yeah. Okay, good point. So like I was saying, this is an example of a consumer resource model. This is not the only consumer resource model. Yeah, in fact, I used to say canonical before in this slide. I was just making sure that I didn't say it. Yeah, so this is just one example. And here, what you're assuming is that all these resources are substitutable, which is that even if you had a lot of one, it's the same as having a little of many. So maybe the thing to imagine in your head is that these are different carbon sources. And it doesn't matter if you have glucose or fructose or this or that. If you really had to worry about essential resources like carbon and nitrogen and iron and so on and so forth, then you could imagine a different consumer resource model that you write down, where instead of this, you sum things, you could have a min function, for instance. And this is also something that people do. Not a lot in cases where there are many species, though, because of some mathematical problems that I don't know the solution to yet. Okay, this is great. Any more questions about? Questions about consumer resource models are great at this point. What they ignore, what they don't ignore. If there are questions about the two assumptions that I talk about with evolution, those assumptions will become more clarified as I go on in my talk. Okay, so assuming there are no more questions, I'll sort of go on. So there are these two assumptions, one that has to do with the central limit theorem, one that has to do with stochasticity, that really are in some sense features when you're doing high-dimensional ecology. They make the calculation easier, but that actually I want to argue that these are exactly the things that become caveats when you're trying to do ecoevolution. And so just to see why this is the case, let's just do a thought experiment together for the rest of this talk. Let's just start with a randomly assembled, highly diverse community, and let's turn on evolution and ask what are the things that we have to sort of worry about. Yeah, and so what is going to happen in the rest of this talk is that there are these 4 boxes. They're going to get filled up as sort of steps of what are going to happen as we're going to start evolving a community. And at some of these steps, we're going to see some problems that are going to come up that I'm going to clarify. And then I'm going to tell you some recent work that we've been doing to help maybe overcome some of these things. And then towards the end, I'll come back and clarify that actually we don't understand much of all of these steps. Okay, so we'll start off. Of course, the first step is that you have a community that you've randomly assembled from somewhere, maybe using the same assumptions of the high-dimensional ecology that I was talking about. And the first thing that maybe you can imagine is going to happen when it starts to evolve is that you have a new mutant strain that sort of arises. Right? And just like I was talking about, this new mutant is not going to be something that's akin to an invader that's coming from outside the community. This is actually going to be something that's very similar to at least one strain, right? Its parent strain. And so that's why this dark green pickle here is a little bit similar to the light green pickle here, where the idea is that you can think of the mutant as an invader, but it's not just any invader, it's a correlated invader. It's correlated in its phenotype with at least one member of the community. Right? And so as soon as this happens, you have to think about this invasion process. And the first thing that you're thinking about when you think about ecoevolution is that this mutant has to beat stochastic drift. So just to sort of get into it, if you studied population genetics, you would have heard of this idea of mutant establishment and how it depends on stochastic dynamics. But if you didn't, here's my 30-second attempt at trying to explain to you the gist of what's going on. Credit to Ben Good for this very nice cartoon and generally an excellent set of lectures on evolutionary dynamics and population genetics. If you don't know about this stuff, I strongly encourage you to read them. They're available online. They're really, really nice. Okay, so here goes my feeble attempt at explaining this idea of establishment. So what we're plotting over here is a plot of frequency versus time. And by frequency, I really mean the frequency of this mutant. That's a fraction of the population that it's in. So a frequency of 0 would mean that the mutant is extinct. A frequency of 1 would mean that it's the only thing that's present in the population. Frequency of half would mean half mutant, half parent. And this is a log scale, just to sort of clarify. So this is log of the frequency. And usually what's going to happen is that your mutant is going to start way down here, right? Early time, low frequency. And because of this low frequency, small population, it's going to have a frequency trajectory that's essentially going to be stochastic. And so these are a few different just cartoon examples of what these trajectories might look like. And the idea is that basically, if this mutant starts off at a small population size, there are cases in which its frequency is essentially doing a biased random walk. And there are cases in which this frequency can go down and hit 0 and then stay at 0 forever. And then there are cases that it can sort of go up stochastically and then keep sort of rising. And one of the central ideas in population genetics is that you can kind of break down this entire process into essentially two different regimes. One in which stochasticity dominates, this is the low frequency regime, and then one in which essentially selection dominates, and this is the high frequency regime. And there is roughly a kind of a scale or a frequency scale at which you roughly switch from a stochasticity dominated regime to a selection dominated regime. And that frequency scale roughly has to do with this number 1 over ns or 1 over 2ns, where n is the total size of your population, and S has to do with something called the selection coefficient that sort of characterizes purely in terms of fitness how much better or worse the sort of mutant is. So if S is positive, the mutant is fitter than the parent. If S is negative, the mutant is less fit than the parent. S equals 0, the mutant has exactly the same fitness as the sort of parent. And so the idea is that if this mutant's frequency manages to make it past this 1 over 2ns, then you can essentially imagine that the mutant is said to be established, and if it's fitter than the parent, then basically it'll just grow essentially deterministically, exponentially, until roughly it hits a frequency of close to 1, right? And if it hits 1, it's set to fix, and when it's fixed, that means it's the only thing that's present in the sort of population. So really, at the end of this process, there are two outcomes. Like a mutant can either go extinct at long time, or it can go and fix and have frequency 1 at long time. And the process of studying population genetics is understanding how this comes about. The central idea is that population genetics tells us what to do when you have a situation where there is just a population with a parent and a mutant. Question? What are the boxes on the plots? You're talking about these boxes? Yeah, I would say don't worry about them for now. If you really went to sort of study it, it turns out there are different regimes that, again, you can sort of break the dynamics into, both in frequency but also in time. So there's a very early time regime, an intermediate time regime, and a long time regime. So these boxes are kind of trying to mark. Yeah, so this is taken from a bigger set of notes, and I just want you maybe not to focus on these boxes. These green boxes here, right? Yeah, so really in these notes, Ben is trying to talk also about the variance in the frequency dynamics, over many different mutants. And so these boxes are trying to, again, characterize what you expect the width of the frequency distribution to be. Sorry if that's what you meant. Thanks. Okay, yeah, so again, for these purposes of this talk, you can kind of ignore them. Okay, so the point that I was trying to make is that population genetics tells us really what to do when there's just a parent and a mutant, that is the low dimensional counterpart. And again, this is the kind of picture that we have. But what we don't have is the analog of what happens when this parent and mutant are embedded instead of just being in a population by themselves, but when they're embedded in a diverse community. And so you can ask the same question of how different do you expect this idea of establishment to be when now they're present in a community, right? And so you can kind of already see that it's not obvious, because this idea of selection now itself depends on the community, because you would expect the mutant and the parent to not just interact with each other, but also with the broader community that exists. And so this is some recent work that we've been doing, which is trying to extend what happens and trying to ask what happens to this picture when you have a complex, high diversity ecological community. So just imagine that you have a mutant and a parent now embedded in a community with many other species, which all have sort of dynamics that you can write down into two parts. You again write down dynamics of the abundance of each of these things into one part that has to do with growth. And so this G sub I is now a catch-all for any growth function that you might write down. It might depend on the abundances of all the other species, of all the different resources that are present. And a second part, which these consumer resource models did not have, which is a noise, and this is essentially demographic noise that just comes from the stochasticity of birth and death. And so the idea is that what we're trying to do is to come up with a general theory of describing the dynamics of this mutant when it's embedded in a diverse community. And when I say general theory, what I mean is that this G of I can be your favorite ecological model. And so, my favorite ecological model, as I laid out, is this one. It's the same one with cross-feeding that I was talking about before. So you can have community members interact through resources. You have different resources that are present. You have different species. And so these species are exactly these species that are present in the community. But these are not the only species that are present in these dynamics. There are two special species. There's the parent and there's the mutant, which you also write dynamics for the same way. I haven't written them down for quantum space. And then essentially what you're trying to do is that your goal is to predict the mutant's frequency dynamics in the soup of this community. And so what I mean by that is that you can write down a frequency, which is the frequency of the mutant, and this is nothing but the relative abundance of the mutant in the parent mutant soup. And your goal is to essentially come up with the dynamics of the frequency of this mutant. Yes?
How do you choose the properties of the mutant? Like, is it a random mutation? Are you choosing specifically, like, this is the kind of mutation that you're encoding?
Great question. So, yeah, so the answer is that you essentially assume a random mutation. But what's special about that mutation, or what's special about the assumption, is that when you're sampling the traits of this mutant, you'll sample them from the parent's traits. But that will be a vector with some correlation rho with the sort of parent. And the larger and larger rho gets, the more and more similar the mutant gets to the parent. Right, so rho equals 0 would mean completely independent mutant and parent. Rho equals 1 would mean the same. And so you want to usually stick to situations in which rho is close to 1.
Akshit?
Yeah.
So a question on when you introduce the mutant, is your pre-existing community at one of its multiple steady states already?
Great question. Yes. So what we're imagining is that you start off a community at sort of steady state. It's going to go to a new steady state in principle when this mutant is going to come up, but you introduce the mutant at the steady state.
And is the original steady state already accounting for demographic noise, or is?
Yeah, great. No, it's not, because then you would imagine that any steady state, if you give long enough time with demographic stochasticity, will be one where there's nothing present.
Thanks.
Yeah. Okay, yeah, so you kind of start off in a situation where not only are you turning on evolution, but you're also turning on the noise right after the communities come up, right? This is not, I'm not saying this is realistic. I'm just trying to say, let's try to add and ask what happens in this situation. Okay, so what we want to write down is the dynamics of the frequency of this mutant. And the technique that we would use to do it is something called dynamical mean field theory. I'll give you a sense of what this is like and how this goes in my tutorial or my class, which is, I believe, next week. And so there are questions about how this goes. Keep them to next week. But I'll just tell you what happens as a result of that now. Okay, so when you do this sort of DMFT, what you sort of learn is that there's effectively one parameter which is the emergent self-inhibition of this mutant that really encodes all of the details of how the community acts on this particular mutant. So the idea is you start from this picture where there's a community, the parent and the mutant, and you essentially want to coarse grain away the community and the effect of the community. And once you do it, you essentially end up with some model describing the frequency of this mutant where there's an extra term whereby the mutant sort of inhibits itself, and that's sort of where all of the effect of the community is. And so when you sort of do that, you end up with dynamics that end up sort of looking like this generically. You have the frequency of the mutant here, which comes in 2 bits, one that has to do with selection, one that has to do with sort of stochasticity. And just to sort of take you through this, there's there are really three different parameters in this kind of effective model. One is S, which is just the fitness advantage of the mutant over its parent. This is similar to classical population genetics, but now it depends on the community. The second is N, which is the effective population size in classical population genetics. In this case, it would be the parent population size in the steady state community into which the mutant comes in. And then the third, which is a new parameter that classical population genetics will not have, is this thing that we call eta, which is precisely this emergent self sort of inhibition that comes in. Right? And it's inside this eta that sits all of the ways in which the community statistically affects the parent. So if you write a different model down, what eta will look like will be different for different models. But essentially, there's going to be some eta that sort of accounts for all the ecological feedback. Yeah, maybe one question here and then the second question over at the back. Yeah.
One question. So one assumption, in addition to the fact that the dynamics without evolution is not stochastic, is that it relaxes to a fixed point, right?
Very good. Yeah, exactly. So there is the steady state assumption that people make. This is not an assumption I was going to attack. So it wasn't a relevant assumption, but very much so. In all of this, there is a caveat, which is that we're only working with communities at steady state. And so if you have communities that don't do that, well, I don't know what to do yet. Yeah. Maybe I'll take the question at the back since there was a hand raised first. Yeah. Yeah, in its traits, which is to say in how it consumes resources. Yeah. Good. Yeah. So that correlation row that I was talking about will be hidden inside this eta. Yeah. Yeah. So all of the gory details of what the community is doing, how things are correlated, they all go into this eta. Maybe Simone, and then the question at the back. Yeah.
So do I understand correctly that you have stochastic dynamics for this, say, gene or mutant? But the rest of the ecosystem evolves.
No, there's stochastic dynamics for everyone.
Everyone is stochastic.
Exactly.
And so how does stochasticity of the rest of the dynamics, I guess it will be absorbed into this parameter?
It gets absorbed, yeah.
Okay.
Yeah. Yes. Do you mind being a little bit louder, please? No, it's not. It's the steady state you start off with is a steady state without the mutant. You then introduce the mutant and then you ask what's going to happen. Okay, and so the answer is that these are the dynamics. If you're interested in what happens to the mutant, this is the equation that describes that. Okay, maybe in the interest of time, I am going to pause questions unless there's a clarifying question. They are the exact same. Sorry, this one. Oh, wow, that's a really poor choice of, sorry, they're completely different, yeah. You just imagine this as sort of W of T. Yeah, thanks. Okay, great, thanks for that. That was really important. Yeah, this eta has nothing to do with this eta. This is just a Wiener process. Okay, good. And so the idea that I was trying to put, which I also put on the last slide, is that the presence of the community, what you learn is essentially manifests as frequency-dependent selection, right? So just if you sort of study the classic Kimura theory of sort of population genetics, what you learn is that the relevant equation for the frequency dynamics is one in which you have constant selection, but the one in the presence of the community is one where you have sort of frequency-dependent selection, and it's negative frequency sort of dependent selection, which has interesting consequences, which I'll get to in a second. And now that you have this, now you can sort of analyze what happens to these sort of dynamics and ask what difference does it really make that my mutant is present in a community as opposed to being present just with the parent. And so the classical way to do this is to talk about something called fixation, which, like I said, has to do with this idea that as you wait a long time, the mutant will displace the parent. And so when you sort of study classical population genetics, you sort of learn of this famous and celebrated Kimura formula that predicts what the fixation probability should look like as a function of the fitness benefit of the mutant, as a sort of function of S. And sort of what you learn is that in the absence of ecology, which is the standard Kimura formula, you learn that there's this sort of nice sigmoidalist relationship between the fitness benefit and the fixation probability. That is, if the mutant is really not as fit as a parent or much less fit, the fixation probability is close to 0. And as its fitness increases, the chances that it fix will get larger and larger, eventually becoming close to 1. And this is indeed what happens in the absence of ecology. This is what happens in the presence of ecology. So this is what we've sort of done here is the blue line over here is sort of a theoretical fit. You can come up with an analytically exact formula for what the fixation probability should now look like with these frequency-dependent selection dynamics. And these little dots with the error bars over here are results from simulations of the entire community dynamics. That is a mutant being present in the community, and you wait long enough to see what happens to this mutant. And you can kind of see, first of all, that there's a good agreement between what the full high-dimensional dynamics of the community are and what we kind of predict with this sort of modified formula. But then you'll also see that there are sort of similarities between what sort of Kimura will predict and what you get in the presence of ecology. So I really will break it up into 3 parts. So first, of course, if the mutant is, if the mutation is quite sort of deleterious, then you expect a match between the two. Essentially, the fixation probability is close to 0. Then is an interesting regime, and it's one in which the mutant is fitter than the parent. Typically, that is, S is greater than 0. And here you see that essentially ecology, or the presence of the community, is strongly suppressing the mutant's ability to fix. Right? So you kind of see this black line over here being a much smoother increase in the fixation probability, whereas this blue line over here with ecology is a much sharper sort of, it's almost switch-like. You go from 0 to 1. Right? And so it's really the presence of the community, we think, that is going to suppress a lot of beneficial mutations. So for instance, look at this guy over here with a fitness benefit about 0.1 or 0.2 or so. Just with the Kimura formula, you would expect a fixation probability of close to half. But then in the presence of the community for these particular parameters, you would expect essentially a very, very low chance of fixation. Right? So it's really a strong effect that you can have. And then, of course, once you have a mutation that gives you a large enough fitness benefit, then, of course, again, you'll start to match what you get with the Kimura formula. There have been people holding their mics at the ready. So if there are questions to ask, yeah.
Is it okay? Okay. So your graph plots a given fitness benefit, let's say S, over the wild type. But can your theory also deal with the situation of, let's say, I specify a distribution of fitness effects? Can you give me probability of fixation? Let's say I say that the mutant sort of follows a distribution of S around the wild type. Can you still calculate fixation probabilities over time?
Okay, so just to understand, when you're talking about distribution of fitness effects, any particular mutant is going to have one of those numbers picked out of that distribution, right? So that number can in principle be any of these.
Yeah.
Right? So if you tell me what number was picked out of the distribution, then I can tell you what the outcome for that mutant should be. Right? But if you're asking me what happens if I throw many, many mutants, all sampled from the same, from some distribution, that I can't do, because there's another assumption here, which is that you're working in what's called the strong selection weak mutation limit, which is that you only put in one mutant at a time.
No, so you can put only one mutant at a time, but if I were to say there's a population and it can mutate by set distribution, you're in the strong selection regime still, so only one mutation will happen, but you don't know which will happen. So can you still use the Poisson's thing that you do in population stuff?
I would say you tell me which S you picked, and I'll tell you what happened.
No, I'm saying there's a probability of picking S's. Can you still get to?
Yeah.
Okay. So that would be, have you done some of that work with the?
No, but we can talk about it after the talk. Yes. It's relative to the parent, yeah. Yeah, so 0 means a mutant is as fit as the parent. Yeah. Yeah. Yeah, it's the fitness of the mutant when put into the community relative to the parent. So this is kind of like the invasion fitness of this mutant. Okay, there are 3 questions. Maybe I'll take all 3. So yeah, go ahead. As long as it's finite, it works.
And is there any trend, for example, that you can see how the strength of this suppression of ecology changes with, say, the size of the community and so on?
Yeah. I don't have it here in this talk, but there's a whole diagram that you can make that maps out how the suppression strength scales with various relevant parameters, including population size, how similar the mutant is to the parent, and so on and so forth.
Depending on correlation with the parent,
Yeah.
only outcomes are no longer extinction or
Good. Yeah. Exactly.
when you say fixation probability, you really have
I essentially mean establishment probability, but just for the purposes of what is happening in these simulations, what you do is that you run the simulation and you wait long enough so that either the parent goes extinct or the mutant goes extinct. If the parent goes extinct, you say the mutant fixed. If the mutant goes extinct, you say the mutant didn't fix. And so this probability of fixation is the fraction of simulations in which the parent went extinct before the mutant. Oh, I mean, there are simulations in which you see transient coexistence. Of course, all coexistence with noise is going to be transient. If I have a few more minutes, I might show you one of those curves. Yeah. If you don't mind, I'm going to go on and try to go through some more of my talk, and then we can have more questions at the end. Not that I don't like questions, but I really do want to say something more than just what happens with population genetics. Okay, so in the 7 minutes that I have left, I'll tell you, and I'll try to tell you a little bit more. But just to get and you know, so just to sort of end this, there's essentially a critical fitness scale of about eta/2, at which you kind of see the switch-like behavior of going from essentially not fixing to essentially fixing. Right? And again, if you only had deterministic dynamics, you would expect this critical fitness to be 0. Right? You would expect a very sharp thing where you wouldn't fix if you were less fit, and you would fix if you were more fit. Okay, and so the kind of low-dimensional outcome or the takeaway from this is that the community, we think, essentially acts as kind of a sharp filter for mutants. So, you know, below this critical mutant fitness eta/2, stochastic barriers are much stronger. So if you want to think about this old picture that we started off with, if you have low mutant fitness, and by low I mean lower than this eta/2, this sort of stochastic barrier is much, much higher than you would expect in the absence of the community. And if you have high mutant fitness, higher than the critical eta/2, then this stochastic barrier is much, much lower. Yeah? Okay, so that was step 2. Of 4, just the idea that this mutant has to beat stochastic drift in order to do anything to the community. But even if it manages to do that, what you now have to worry about is you have to worry about the dynamics of this correlated strain in a highly diverse community. And so there also it turns out that there's lots of counterintuitive dynamics that you would observe compared with what you would expect from the simple high-dimensional ecology picture that you might get. And so just to give you a taste of this, not that I have too much time to go into this in detail, the kind of random matrix arguments or the central limit theorem arguments that I was talking about, these are the ones that start to break as soon as you have a mutant that is strongly correlated with the parent, right? Essentially in your sort of random matrix, you know, you have 2 rows that are very, very correlated. And so when you think about this process of a correlated invader coming in and invading the community, what you now have to do is you now have to come up with a general theory of invasions, one that can incorporate not just invaders that come from outside of the community, but ones that incorporate invaders that come from inside the community. And so in this sort of manuscript over here, that's sort of what we tried to do. again, the general idea is that it's a general theory because it works in principle for any ecological model. And by any, I mean it's one where you reach a steady state. Yeah, and so here also there are some counterintuitive lessons that we see in sort of high dimensions. And one of them is that despite the strong similarity or the strong correlation between the mutant and the parent, what you can often find is that the mutant and parent can often coexist. And again, if you live in this standard population genetics world, what you always expect is one to outcompete the other, never for this coexistence to occur. But now maybe you can kind of already see with this kind of negative frequency dependent selection, you can expect in principle some sort of coexistence between the parent and mutant to occur. And what I mean by this is that when you actually do simulations in the presence of communities, what you generically find is that it's very, very often that you see parents and mutants coexist. It's not just a rare event. And you might ask yourself, okay, if the community is very, very packed this way, that it has many, many species, something you might expect needs to go extinct. And so what might that be? Like I said, your naive assumption would be if the mutant comes in, it would knock out the thing that would be closest to it. But actually what happens is that often there is a very metabolically distant species, not distant in space, but distant in sort of phenotypic space that is actually knocked out. So just the two things that I want to tell you here is, again, just to sort of expand on this idea of coexistence, and second on this idea of what is knocked out. So despite this strong similarity, what you can often observe are the mutant and parent coexisting. So this is one example of what these dynamics would look like. The blue over here is the mutant, the black over here is the parent, and the grays over here, many of them are just all the different members of the community, and just these are sort of highlighted for you to see. And you can kind of observe that there's a long time for which this parent and mutant can coexist, but eventually if you wait long enough, one of them will of course go extinct. And so this coexistence, however transient it might be, can only happen because of the community. There's no generic coexistence that you would expect just with the parent and the mutant. And then the second thing is that this idea of this distant species being knocked out. So sort of what you generally learn from this theory, that it's the low abundance strains, regardless of how similar or different they are to the mutant, it's the low abundance strains that are most likely to be knocked out. And so, one way to see this is that you can plot a distribution of the extinction probability as a function of the abundances of the different community strains that are present. And it looks roughly exponential, no matter whether the community is young, which is that it's been evolved only for one step, or whether it's quite mature, which is that you do this evolutionary process where new mutants come up and can fix and can cause other things to go extinct, and you do it many times. You kind of always see this roughly exponential-ish distribution of the extinction probability as a function of the abundance. And there's no correlation between the similarity between the mutant and what goes extinct. Okay, and so the takeaway here that I want to make is that the dynamics of mutants can be quite different in the presence of a community. And just now in the last minute, what I'll say is that when you sort of do all of these three steps, all you do is you kind of complete one step of the evolutionary process, so to speak, right? You have one mutant that came up, beat some stochastic barrier, managed to establish itself in the community, maybe knock something else out. But over a long time, you now have to worry about many steps of this process, so-called long-term evolution, in which case you might start to get communities that look very different from what you sort of started with. And so here also some very interesting things can happen. And if you're interested in sort of more of this, I encourage you to also look at sort of Aditya and Daniel's paper. Aditya is right here in the audience, so I encourage you to catch him and ask him about this very nice work that they've done. And so what you typically find is that as you have more and more mutants, you reach a kind of a strongly correlated state. And what I mean by this is that you end up with a community in which there are kind of clusters of similar strains. So you can have clusters of these green guys, clusters of these red guys. And again, I don't mean spatial clusters, I mean clusters in sort of phenotype space. You still observe that the diversity of the community appears to be evolutionarily stable. That is, the whole community doesn't always kind of collapse, but it's sort of very different in how it's structured, in particular because of these correlations. And one of the sort of consequences of that is that some of the predictions that you could make from this high-dimensional ecology and random matrix theory, for instance, one of the more celebrated ones is this idea of what the species abundance distribution is. That starts to look very different for evolved communities as opposed to assembled communities. And so this is, again, just a cartoon to show you how different they are. And if you're interested, we can sort of go into it. But this kind of has implications for macroscopic patterns, because this is often a thing that is relatively measurable in communities. Okay, so what I'll say, and maybe if I have 30 seconds, is that by now we've gone through all of these 4 steps, and maybe what you can observe is that there's a lot of opportunities and steps where we don't understand things. And so really all of this stuff, I would say again, sort of challenges high-dimensional ecology, and it's not sort of well understood. And this, I think, represents a tremendous opportunity for a distinct theory of ecoevolution in sort of high dimensions. So I had a whole slide about maybe questions and gaps. Maybe we can bring them up in the discussion, but in the interest of time, I'll just end with saying that I hope what I've done today is sort of try and give you a glimpse into how incorporating evolution in this high diversity community ecology is a challenge, and how maybe it's not going to be solved by simple extension of all of these high dimensional ecology frameworks, because it breaks some of the central assumptions. And instead, it's sort of tremendous opportunity to do new theory, and perhaps ones that incorporate genes, organisms, and the environment together. Thank you for your time. I'm happy to take more questions and also into the coffee break. Yeah.
Very nice talk. Couple of questions. One is a quick one. The slide you showed where low abundance mutants tend to get knocked out. That goes back to Wright Fisher and all that, right? It's generally true, or is there something
Goes back to Wright Fisher. So, and Wright Fisher, as far as I understand, just neutral Wright Fisher is everyone is essentially the same.
Yes, yes. So
and
it's
then there it stands to argue that if you're low abundance, you're more likely to be knocked out because you experience noise more strongly. But this is a situation in which there's actually selective differences between different species. So some are more fit, some are less fit.
So you could have low abundance species in a more simple model that actually gains, but here you're saying it gets.
Yeah, it just gets killed. Yeah. The general idea is every time you introduce any invader into this community, is going to act essentially as a perturbation to the effective environment that the community experiences. And so every strain is going to see the effect of that being some shake up to its abundance. And so essentially the ones that are lowest abundance experience, have a lot more to lose with that shake up. The ones that are higher abundance tend to lose a lot less.
So next and last question is, in terms of validating the theory, is it feasible to do agent-based models, simulations to simulate large ecosystems and diverse ecosystems and so on? Do you think it's a possibility?
Sorry, I didn't follow this. So were you talking about validation using agent-based models or using data?
Using agent, or cross-comparison using agent-based models to compare the parameters.
Yeah, so the stuff that I showed you, like for instance, some of the stuff for this population genetics work was not through agent-based models, but it was through exact simulations of this entire, full ecological dynamics that I was talking about. And really, since that's the process that we're talking about here, and there's no space per se, you don't really need to do agent-based modeling here. If you, I guess, were really worried about individual level things, then you would, but that's not what we did here. Yeah.
So you said that there's not, like if you introduce a closely related mutant, it doesn't kick out its parent, but surely if I make it close enough to the parent, then it must.
Yeah, exactly. So one of the other things that I didn't show you is that what we found is that, so you can ask what happens as a function of how closely related the parent is to the mutant. And there seems to be a critical correlation, which is like in most of the things that we worked on, like 0.999 something. If you're more correlated than that, then it definitely does matter. Then essentially there's only one thing that survives. But if you're less correlated than that critical correlation, then you don't see this.
But is it not true that the probability of coexisting sort of scales with the the difference between as that goes to zero, then probably you're not having coexistence because you have the noise.
Yeah, I mean, so but even if you didn't have the noise, yeah, so indeed what I thought was that there would be something smooth with the probability of coexistence as a function of how similar you are. But at least what we find right now in sort of numerics is that that doesn't seem to be the case. Maybe I'm answering a different question to what you're actually asking, and we can, yeah. Yeah.
Question. So I don't know if you said it, maybe I lost it, but so you get this single parameter eta from this dynamic mean field.
Yeah.
But I would like to get a sense of how big is eta in like say in a potentially realistic situation, and what does it depend on?
How big eta is in potentially realistic situation? Yeah. Okay.
In the same vein, why is eta always positive?
Okay. Why is eta always positive? Okay, I mean, I guess first the question is eta always positive? And yes, it is. And all of the soup of what eta constitutes, yeah, it definitely should be positive. Why it's positive? I think I don't know a succinct answer to this question. I would say generally what you see in most ecological models is, regardless of whether there's cooperation or competition, what essentially you see between all strains in a community is some effective competition in the effective environment that they all create for themselves. And so what I'd say is that in the end, because the community is suppressive towards the mutant's effect on itself as mediated by the community also ends up being suppressive. This is perhaps not the most convincing answer that I can give you, but this is sort of
relative, sort of with respect to the parent.
Yeah.
In dimensions,
yeah,
such that the community actually helps the mutant rather than with respect to the parent.
Yeah.
F is frequency with respect to the parent.
Yeah, I know that's true, and certainly there could be situations in which you kind of design mutants that for which this is true. I guess what I was talking about here are sort of random mutants, which are mutants where you sample all of these phenotypic parameters randomly. And I think just because there are many different resources, what ends up happening is that systematically when you pull things randomly, this turns out not to be the case. But certainly you can design mutants for which this is exactly the case. And then this might not be true. But then again, this only applies for random mutants. To go back to answering the question about how large eta is, I don't know. But what I'll say is when we saw this kind of diagram, what we became very excited about was trying to test some of this in data. And so what we thought we would do is we would just look at kind of neutral mutants and try to see whether they were more or less likely to fix in a community versus not a community. And what we quickly realized, and I don't know if this is actually true, is that even the Kimura formula, this black curve over here that is celebrated, that people like a lot, that people infer a lot of parameters, as far as I know, there is no empirical evidence for the Kimura formula. So, which is to say that not only did we not understand how we could test it, we didn't understand how you could test the simpler version. So if you have thoughts on how to do that, I would be very curious to chat. But because we kind of don't have a sense of this, I would say I don't know in terms of numbers how large we expect any of these things to be.
But I would say like if you have to gain your model reasonable numbers.
Okay, so just from the model, I mean, I guess it really depends on how you tune the parameters, but typically, like if you have S's of order 0.1, you could have etas of order 0.1-ish as well. Yeah.
I have a quick question about this this plot. I mean, what you're testing here is what happens over very long times after one of the two goes extinct. So this effect is basically, I mean, the intuition behind the blue curve is that they go to a population which is S over eta, and then the one with the lower population has a higher probability, and that's why you have the transition. Okay. So really they coexist for a long time, and then, okay.
Yeah, and actually the idea is if you sort of start to think about a potential well-like picture, there's a well sitting at eta over 2. So there's a well sitting at S over 8 eta. And really the question is, will you escape this well on the left or on the right? And the probability of doing that depends on whether S is larger than eta over 2 or not.
And this effect is driven by the arrival of another mutant that drives extinction in the community?
No.
No, not of the mutant of the same pair. Something else, like that drives a change in the community that then drives the extinction between these two.
In this picture,
Or
no,
is noise?
it's just noise.
Ah, okay.
That's what I meant by strong selection weak mutation. You wait for either the parent or mutant to go extinct before you will do anything else. So it's really not something else coming in. It's really parent and mutant are sitting happy. If you turn off noise at that point, they will stay happy forever. But it's just, you know, this potential well in which they're sitting somehow coexisting is shallow enough that you can get out of it.